Now we're going to talk about the evolution of the genetic code. And this is a beautiful example that shows that all animals, all plants, all living organisms share the same genetic code. And actually, there are very small differences between all these very diverse group of living organisms. And one beautiful example is that when you take proteins that are fluorescent of jellyfish, you are able to put it into different animals. And this is why we have on different, even in bacteria. And this is why we have all this revolution with fluorescent proteins, because you can take a gene from here, a section of DNA, and insert it and paste it in all these organisms. And now, as we have the same genetic code, the protein is going to be transcribed and then translated. And as a result, we will have all these animals that are fluorescent. So remember that the genetic code is nearly universal and is shared by the simplest bacteria to the most complex animals, and genes can be transcribed and translated after being transplanted from one to another species. And this is exactly what happened here. As I said to you, we took a gene from a jellyfish and we put it in a diverse group of animals, and now all of them are fluorescent, and this is how we can see that the cells move, or we can do many experiments and track what is happening with each one of these organisms at the cellular level. Now, we already gave you a very brief introduction about transcription and translation, about what are the difference between prokaryotes and eukaryotes. Then we talk about what is the genetic code. In this slide, I will talk about how point mutations can affect the protein structure and function. And I know that you have heard about mutations, maybe from movies like this one. However, I will focus on the scientific definition of a mutation, that are changes in the genetic material of a cell of a virus. So I'm sure that you now are an expert and you have seen in the news that the coronavirus is changing, because viruses are able to change much faster than most of the other organisms. And in particular, there is a type of mutation that are called point mutations, that are chemical changes in just one base pair of a gene. So here in this diagram, I just wanted to show you the difference between an original sequence and a sequence that have a point mutation. And if you see, all these sections of this diagram are similar, except for this and this one. Yeah, so if you compare, here you have a T, here you will have a T. However, here the T is turned into a C. And this will have implications to the protein, because this, as we showed before, it can create a different amino acid, and it can change the structure and also can change the function of a protein. So the change of a single nucleotide in the DNA of a template site can lead to the production of an abnormal protein. So here, for example, all the different amino acids are still the same. However, well, in this case, this one will be different. Now I'm going to give you an example, and it's the mutant hemoglobin DNA, and this will be the template strand, and I want that you get this messenger RNA, and I just remind you this will be the complementarity of bases, and in particular this could be helpful, this uracil. So stop the video and try to find the answer. So this is the answer. The C will be complementary to the G, so this is how you get this one. The A will be complementary to the U, and finally the T will be complementary to the A, although here it will be replaced by a U, but this is the sequence. And remember, the template strand will always be go from 3' to 5', and your codon to messenger RNA always will go from 5' to 3'. Now I want that you again stop the video to get the protein, but I want to give you this as a clue. Okay, I imagine that you already found the answer. So this is the sickle cell hemoglobin, and here it will be this amino acid. And how you get this amino acid, then maybe you already studied this in high school. In case that you didn't do it, remember this will be the first messenger RNA base from the 5', and this will be the second messenger RNA, and this one will be the third messenger RNA base from the 3'. So we have the first one will be G. So here it will be G because this is the first one. So it can be in any of these ones. Any of this one will be. Now the second one will be U. So it will be here. So it could be this one, and it will be the intersection. So it will be one of these. And finally, so it will be one of this, and the third one. And the third one will be an A. So it will be specifically this one. However, if you see only with the two first ones, as all the other four are code for the same amino acid, because the genetic code is redundant, we could have already know which one was the correct one. So I hope that that was clear. Again, you go first to this one, then you get the second one, and then you move to the third base. And this is how you get the intersection here, and later you get this A. And if you have a question about this, just let me know and we can do more exercises in the classroom. Now let's talk a little bit more about point mutations, and they can be divided into two general categories. We already talked about the first general category, that is are the base pair substitutions. So let's make an analogy with a sentence. Here, if you replace the D for an M, so the red dog beat the tan cat, now instead of dog you will get mog, and this result in a nonsense. The same thing will happen with the DNA. This is the example again. You have a change, and as a result you have a different protein. However, this is small change. Let's see what happened with the other category of point mutation. And why I wanted to show you that example, because we have been talking about substitutions, and in the previous example of a substitution, we found that there was a different DNA sequence, so as a result there could be a different protein, but it's not always the case. In this case, for example, you got a silent mutation that there was no effect on the amino acid because of the redundancy of the genetic code. Now we're going to talk about missense mutation and nonsense mutation. And to do that, I will have this diagram that I want to show you. Here you have a change in the T, and here you have a change to an A. So here is the wild type, and here you have the mutant. And if you see here, although you still have an amino acid, there is no change in the frame. There was a change in the type of amino acid. So you still keep making amino acids, but it's not the right amino acid. So this is a missense mutation. You produce a change in the amino acid, and this is the main difference. On the other hand, the nonsense mutation produces a more dramatic change. Here, you also have a point mutation, in this case. A T is replaced again by an A, but instead of producing another amino acid, you produce a stop codon, so there is no more translation. And this is terrible. Why? Because, well, here you have a different protein. You still have 1, 2, 3, and 4 amino acids. What would be the effect? It could not have a major effect. However, when you have a stop codon, you miss all these amino acids. So you have these 2 amino acids, even you have 3 amino acids out of 4. So the nonsense mutation changed the amino acid codon into a stop codon, very important. And another important thing is that nearly always lead to a non-functional protein. So the missense mutation produces a change that could lead to a non-functional protein. However, when you have a nonsense mutation, you start missing, you can miss a large portion of the protein, and this can cause an that is almost always a non-functional. Now we're going to talk about base pair insertions. And here you can see the DNA sequence and here the amino acid sequence. Here you have a normal string with 1, 2, and 3 codons, and here 1 and 2 and 3 amino acids. Here you will have an insertion of 1 nucleotide, and on the other hand, here you will have 2 more nucleotides. So the previous one here plus 2 more nucleotides here. So let's see what happened. So in the insertion of the mutation, here you're going to have a change in the frame. So you move all of them 1 nucleotide forward, and as a result, here you are not going to have a 4 codon finish. So you will have the same amino acid in the beginning, but the other 2 will be different. And the 4th one is not even complete. Now in this case, in addition to adding 1 from the previous one, we add 2 more, so we have an extra codon. So if you see here, still we have the same one because it's the same first codon. However, this one will be different. And as you, in this case, as you use these 2 for here, from the codon 2 to the codon 3, and this one to the codon 3 to this to be the end of the codon 3, now you have a completely different protein, and the same will happen in the 4 protein. So as you can see, while a point mutation that produces a substitution can have a change when you have an insertion of a nucleotide or a deletion of a nucleotide, there will be much more drastic changes. So to remember, insertion and deletions are addition or losses of a nucleotide pair in a gene, and these mutations have a very terrible effect on the resulting protein more often than substitution do. And why? Because insertions or deletions of a nucleotide may alter the reading frame and produce a change in the frame which can change all the amino acids of a protein. And this is the example with the base pair deletion. This is the normal one and this is the deletion one. And here you can see that when you lose this one, there will be a change in the frame, and now you will have very different amino acids from now on. In this final section, I want that you identified some of the examples of mutation that we just studied. So I want that you try to identify if there are some nonsense mutation, some missense mutation, or type of point mutation. So let's start. Now we're going to do an example of a point mutation. Here's your wild type, your DNA template strand. Here it will be your complementary strand, your messenger RNA, your protein. Here you have your initial amino acid, and here your stop codon. So I want that you see what happened here. What happened if you replace an A, you have an A instead of a G. So you're going to change this one, and this one, and you will see what happened here. So we'll stop the video and you can find the answer. So here you can see that as you change an A here, and the complementary will be a T. So this will be a difference in your complementary strand. Here you will have a U because the complementary, well, G is the complementary of C. Now, in the case of A, the complementary will be U in the RNA. And now if you go and if you see if there was a difference, you found that actually there was no difference, because remember the genetic code is redundant. And here you find this is an example of a silent mutation because there was no effect on the amino acid sequence. So in this case, this point mutation didn't have an effect. Here you have another exercise, and you have again all the information here. And here the changes that it was a T is replacing a C. So here all this one will change. So try to find the answer. So please stop the video and try to find the answer. And the answer is the following. The T now will produce an A instead of a G. And here instead of G, you will have an A. And again, here you will have a different one. So instead of glycine, you have serine. And as a result of that, you will have a missense mutation. Now another exercise. Let's see what happened. Again, the same dynamic, but now instead of an A, you have an A instead of a T. So all of this one will change. So please stop the video and try to answer this question. So here you will have a T. Here you will have a U. And here you will get a stop codon. So this will be a nonsense mutation. Now this is another exercise. Here you have an extra A. So I want that you tell me what you will find. Here you will get a T. Here you will have an extra U. And here you will have a stop codon. So there is a frame shift causing an immediate nonsense mutation due to a one base pair insertion. Now let's try to solve this one. Now you are missing an A. And as a result, you're missing here a U. Here you change, and now you have leucine and alanine. And as a result, there was a frame shift causing an extensive missense of one base pair deletion. Now let's go and do this exercise. So now you're missing an entire codon. Stop the video again and try to answer the question. So this will be your complementary strand. This will be your messenger RNA. Here you will get a stop codon, and there will be no frame shift, but one amino acid is missing due to the 3 base pair deletion.